Right. Welcome all of you for Jamie's PhD defense. I just want to start saying that Jamie joined my lab in 2018, early 2018, when I was just 1.5 year old in UCLA. And that time also first year I didn't have any students. So Jamie were virtually the first students I hired. That time she was an undergrad, second year undergrad students, and she wanted to have some research and just choose my lab for whatever reason, I don't know, but I'm grateful that she came. And then she wanted to do some research on environmental health and air quality. And I'm not an air quality person. And that time, microplastic is something was I know is related to that. And my colleague Sujit Ravi, who is here, he's a air quality person. So we thought, why not we do research on microplastic? Because I was not interested in microplastic. I thought it's the same thing as colloid, something that I did in PhD, and I feel like there was no new science to do here. Um, but I'm glad that both Jamie and Sujit convinced me, and I also wanted to learn more on air quality. And so without Jamie, the microplastic research would not have been here. So she's the one who started it, and she developed a new technique using smartphone how to analyze microplastic, and then that opened up a lot of things, and other students benefited from that and got PhD before her. So it's pretty much she's the one who why microplastic research is in my lab. So I'm grateful, Jamie. And so again, this is all her research in last 5 years or so. She is going to present only section of her PhD because, interest of time. So again, if you have any questions, you can put on chat window. You can also mute your audio just in case, there is a background noise. But if you have burning question, feel free to stop and ask Jamie. Thank you all for being here, and we should start. This is Jamie for all of you now.
Awesome. Thank you for that very nice introduction, Sanjay. Yeah, for those of you who don't know me in the audience, my name is technically Donna Jamie Leonard, but I go by Jamie. Again, I'd like to thank my community all for being here and being so flexible with the remote access due to health reasons. I plan to present my PhD thesis today, and this body of work is titled Airborne Microplastics, Exposure Factors, and Resuspension Mechanisms. A little bit about me, actually. I've only ever been a UCLA Bruin, so I started at UCLA in 2016, got my bachelor's from the IUS school in environmental health and engineering in 2020, then moved to this department in 2021, got my master's, and then now it's 2024, and fingers crossed I got my PhD in civil engineering. During this long time, I've had a couple notable achievements, one of which was being awarded the NSF GRFP in 2020, which is part of the reason why I ended up in grad school. And actually also got the GSA Service Award for contribution due to being president of the Engineering Graduate Student Association for the last 2 years. It's been one of the biggest blessings of this PhD's service. And having things to do outside of research, I guess. And then speaking of research, I've published 12 journal articles, 4 of which are lead author, and I have an additional 4 in preparation and review as lead, some of which I will be discussing today. As Sanjay kind of briefly introduced, I started in 2017 or 2018 in his lab. At the time, I had no idea I was going to go to grad school, but working with kind of all the remarkable master's and graduate students and other undergraduate students at the time, I ended up really wanting to go. Thanks to his mentorship, I had a good basis in research, applied for the NSF, got that, and ended up here I am today. I really attribute that all to him. He's been an incredible mentor. And I'm proud to share that the next step, despite kind of waffling between academia and industry, I'll be working for EPA Region 9 as an air quality scientist. So let's jump right into the work itself now. So plastics come from everywhere. So anytime you eat anything from the grocery store, it usually comes in a plastic container. You brushed your teeth in the morning this morning, fingers crossed, you likely use plastic bristles. Pens you might be taking notes on this presentation on usually have plastic components. Your phone, plastic components. If any of you are wearing, polyester fleece, I can't see it because we're remote, but also sheds polyester. So We use all these plastics in our daily life, and while plastics are produced as microplastics as microbeads and other kind of primary microplastics, all this kind of plastic products that we use in daily life actually also can degrade in the environment and become secondary microplastics. And what are microplastics? They're usually created from plastic products like what I just discussed, and they have a lot of kind of unique properties. Part of them makes them such a valuable consumer product, but it also makes them such a persistent pollutant. So microplastics are classed as greater than 100 nanometers or smaller than 5 micrometers. They come in a wide variety of shapes and sizes, spheres, fragments, fibers. Oftentimes they're created from the breakdown of, plastic trash. So that comes in, kind of funky shapes. They have a density that usually varies between 0.9 and 1.4 grams per centimeter cubed, which is fairly low density. And there's many common, they're made up of many different types of polymers, but some of the notable ones are polyethylene, polypropylene, polystyrene, and polyethylene terephthalate. And we know that microplastics are everywhere in the environment, right? So they're created, we use them, they break down in the environment, and they shed literally everywhere. I've been lucky enough in my PhD to kind of look at plastics within almost every environmental compartment. On this left side is from a study where I looked at microplastics in the depth of the ocean core. Sorry, I'm trying to find my mouse. At the depth of ocean core down to 20 centimeters. In this review paper I was part of, looking at 196 studies from 49 countries. While we found them in all sorts of waterways, one of the more interesting takeaways from this is they were actually found the highest in glacier and snow. And while typically we think of microplastics being dispersed in the environment through water, water moves downwards from glaciers. They don't go back up. So they're not necessarily connected in that way. And that means that air transport or wind as a vector of microplastics could be a pretty significant vector. And there's a lot of literature to kind of support this. So transport of microplastics in the environment through soil, through water, through air, it's really, really popular. Here's some kind of somewhat recent news articles. They're found at the top of Mount Everest, in remote mountain catchments. They're in the bottom of our oceans, like I've discovered. They're in our soils. They come from car tires, they come from clothes, they come from everything. And ultimately, they're all around us, and wind is a pretty significant vector. And why should we care? A is because microplastics are actually found in our body, and a lot of articles have come out recently. They're in our blood, our lung tissue, deep in our lung tissue actually, intestinal tract, in our reproductive organs, in all our organs really, our renal systems. They're in our vascular systems, our hearts and our arteries and things like that. So they're everywhere in our body, and why should we care about that is, while they haven't traditionally been correlated with actual adverse health effects, most recently they were. So in March, they were correlated with atheromas and adverse cardiovascular events. That's just the tip of the iceberg. There are very limited studies on human health, but I'm sure that more are to come. Like any, popular pollutant that's come out and, had health effects like asbestos or DDT, plastic could be one of those. So it's important for us to try to understand microplastics. Where are they? How do they move? How are we exposed to them? And they get into our body through two main exposure pathways. One is ingestion, eating fish, eating plants where they're uptook into their roots, paper cups, plastic cutting boards, tea bags, things like that. Those can be controlled by trying to, moderate which mediums you're being exposed to, but you are exposed to it through inhalation, right? Some studies have found that you might inhale up to 7,000 particles at home, even indoors. You can't necessarily manage inhalation unless you want to wear an N95 mask for the rest of your life, but we all did that for 3 years and we weren't super happy about that. So understanding atmospheric emissions, atmospheric quantities of microplastics, these are really important topics. An atmospheric emission of microplastics, like many colloids, kind of follow traditional colloid theory. So they get into the atmosphere through mechanisms like siltation, through direct suspension, through the breakdown of emulgations of particles and releasing microplastics into the atmosphere. What they're actually a function of, really, though, is how much is on the Earth's surface, so how much is there that can move into the atmosphere, and what environmental conditions affect their emission. We often think of that as wind, but as I'll break down throughout this presentation, there's many other factors that affect the emission of traditional particulate pollutants and may affect the emission of microplastics as well. So I've talked a lot about what we do know about atmospheric microplastics, but what don't we know about atmospheric microplastics? We don't know whether microplastics are more susceptible to being emitted to the atmosphere than other particulate matter. We don't know what microplastic properties may make them more prone to emission. We don't know what factors could influence exposure to airborne microplastics, so this could be environmental factors like UV exposure, climate, land use, humidity, or this could be population characteristics, also like regional climate or socioeconomic status, proximity to sources, things like that. And we don't know to what extent do these factors influence emission or exposure. And that's essentially what this body of work solves to answer all of these questions. So the objective of my thesis is to kind of improve our mechanistic understanding of microplastic emissions into the atmosphere, as well as the factors that could influence human exposure to airborne microplastics. Let's kind of walk through my chapters, I move all the way from terrestrial systems, where I'm part of papers actually looking at microplastics in soils or microplastics in stormwater systems, and how they may be affected by land use, like industry and wastewater treatment and landfills. I looked at transport of water all the way down to the depth of the ocean and ocean core sediments, which are truly thought of as the ultimate sink of microplastics. But the bulk of my work, and what I think is the most interesting part of my work, is looking at airborne microplastics, verifying their emission, verifying their preferential emission, looking at what factors cause that emission, and looking at the forces that control that. So let's kind of walk through the scope of my work individually. My first 3 chapters I'm not presenting on today through the interest of time, but I'll just kind of introduce them. Chapter 2 looks at the effect of land use and climate on the spatial distribution of airborne microplastics. So kind of looking at this graphic, I look at climate, regional climate, as well as the land use for global studies, and I try to correlate whether higher microplastic concentrations were found with any of those factors. Chapter 3, I took a depth profile of microplastics in ocean core sediments from the Gulf of Mexico down to 20 centimeters. I looked at that depth profile and tried to draw conclusions about historic deposition. Chapter 4 looked at the variability of airborne microplastics on leaves. Urban canopy has been used to monitor particulate pollution a large number of times. I kind of wanted to see if the same could be done for microplastics, so I looked at a bunch of different factors for leaves, like their height, their type, their hydrophobicity, and ultimately just found that there's a lot of challenges, so we don't use that method anymore. Chapter 5, 6, and 7 I will be presenting in depth today. So Chapter 5 is on the effect of land use, like industry, remote, rural, these sort of things, and socioeconomic factors such as poverty or being classed as a disadvantaged community on the distribution of airborne microplastics. And then Chapter 6 and 7, Chapter 6 verifies the preferential emission of microplastics using both a theoretical framework and experimental results from a wind tunnel experiment. And then Chapter 7 actually looks at UV exposure and humidity effects on the emission potential of microplastics by kind of zooming in and looking at that and quantifying the adhesion force between microplastics and soil. Before I get into all the chapters, Dr. Mohanty kind of introduced this, but we use a method throughout all of these chapters that I'm just going to go over once. It was actually the topic of my thesis in 2021, and it was published in 2022, Rapid Quantification of Microplastics Using a Smartphone. We have a Nokia with a 3D printed attachment that has like emission lens, some LEDs, external lens. We can take filters with plastics that have fluorescence, zoom into them, and we have an automatic MATLAB algorithm that can count them. So how does the extraction of microplastics work? We can take soil samples or water samples. We use density stratification using a Ki solution of 1.6 grams per centimeter cubed, which is much heavier than the density of most plastics. We vacuum filtrate that. We stain the dye using now red, which confers selective fluorescence on plastic. We can take a picture using my smartphone setup. Here on the right side, you can see it. We have a MATLAB algorithm that counts that. This method has been used for all the papers in my thesis, as well as a lot of you were here for Dr. Kutnick's thesis and some of Dr. Borthaker's thesis. So it's a method that's been used many times, and we're lucky to publish a lot with it. Without further ado, let's jump right into chapter 5. Chapter 5 tries to answer whether socioeconomic and land use factors correlate with microplastics in deposited dust at schools in Los Angeles. So we have microplastic pollution. I look at land use like remote, rural, and industrial sites, as well as socioeconomic characteristics like poverty and disadvantaged communities. The motivation for this study is whether we can apply our understanding of particulate exposure to predict microplastic exposure. So we have many sources of particulate pollution in urban areas, and many of those factors have already been proven through lengthy literature to correlate with higher exposure. Some of those factors are proximity to sources, so proximity to freeways, industrial sites, landfills, wastewater treatment plants. Some of those factors include socioeconomic factors, like poverty, like a percent of a vulnerable population, such as the elderly or children, household size, income level, the percent of respiratory disease or heart disease in a region. So all these factors are already known to correlate with particulate matter, and microplastics are just a subset of any other type of atmospheric particulate matter, but it's unknown whether they follow similar trends. So this chapter is the first to ever try to explore whether this is true. So we took deposited atmospheric dusts from 66 sites in Los Angeles. They were school sites and also sites from the California Air Resources Board to try to kind of have the same distribution as the already existing regulatory board in this region. This map kind of displays the spread of our sites, as well as the general concentration. They go from 0 to 227 particles per gram. We examined 5 variables. So socioeconomic variables we examined were the poverty in that region, as well as the disadvantaged community status at that site. We looked at the distance to landfills, the distance to wastewater treatment plants, and then the distance to industrial sites. The goal of this chapter, of course, is to see, can we predict the spatial distribution of airborne microplastics with any of these already proven to be relevant variables, and produce kind of similar maps as this one, where you can see poverty and also predict perhaps atmospheric microplastics within that region. The results of this study, looking at poverty, which was our strongest variables, poverty has been associated with particulate matter many, many times. Using t-tests, we found no or limited association with poverty. So this map on the left, here we have dots with increasing size as the pollution level in that site increased. And the darker color blue means higher poverty. So we can see a couple, darker, larger blue sites. When we did t-tests and produce a box plot, we did not find a statistically significant correlation with poverty. However, we then did further statistics. So I've kind of put this old graph up in the left corner just so you can keep it in mind. We then did a Fisher's test. Fisher's tests usually have to kind of establish a baseline or something that you're testing. So we kind of tested this full range. So here on the left side, you can see that graph. And you can see we have a double peak, but the larger peak happens here at around 60% poverty. And over here on the right side, when we tested that against MP threshold using the Fisher's test, we can see a jump out here at around 60% poverty and about 30 particles per gram. So there's a weak association between poverty that's larger than 60% and microplastic concentrations being higher than 30 particles per gram. And that's actually the most correlation we found with any variable. So Fisher's test did not reveal any association with proximity to any of the other source variables. So here we have poverty. You can see what that looks like at around 30 microplastics. You can see there is a significance with 60% poverty. But for the other pollutants, for proximity to industry, proximity to landfills, proximity to wastewater treatment plants, or designation as a green zone, which in Los Angeles County, green zone is kind of the jargon That's what we call disadvantaged census tracts in this county. And we can see that we didn't find any association with those source variables. Breaking that down individually, here we have green zone districts, so kind of like what I just talked about. Anywhere you see a green census tract means that LA County has marked that, designated that a disadvantaged community. And all the orange dots are our sample sites within this region. When we plotted the microplastic concentrations, whether it was a green zone or not a green zone, we did not find a statistical significant difference with microplastics, so they're not necessarily exposed to more microplastics if you're living in a disadvantaged community or a green zone. This trend continues on. Here on this left side, anywhere you see a black cross is a landfill site. There's a huge number of landfills within LA County. We have a huge population. Again, the orange dots represent the samples taken for this project. And when we plotted that using a t-test and a box plot here on the right side, you can see proximity to landfills may not expose populations to significantly more microplastics. We did not find a statistically significant difference. Again, that could be due to the relatively high standard deviation. Proximity to wastewater treatment plants also did not expose populations to significantly more microplastics. Here on this left side, you have the map. All those little green triangles are wastewater treatment plants within Los Angeles County. The orange are sample sites. We use a distance of 5 kilometers just because there are a rare number of wastewater treatment plants within Los Angeles County. Both 2 kilometers and 5 kilometers have existing literature to establish those as relevant guidelines for distance. For industrial sites, here all the little purple manufacturing sites within Los Angeles County are visualized. And when we plotted that box plot, again, we did not find a statistically significant difference. So proximity to industrial sites did not necessarily expose populations to significantly more airborne microplastics. So what is kind of the take-home message of this chapter? The first is that only poverty exhibited a significant relationship with microplastic concentrations over 30 particles per gram, albeit that association was weak and not necessarily linearly proportional, but still, there is a correlation. The spatial distribution of deposited microplastics did not correlate with spatial variations in other socioeconomic status like designation as a green zone, nor with proximity to sources. This is unlike traditional particulate matter, and this is the first study of its kind to kind of explore that. So I'm sure future studies will explore that relationship more and see if there is actually a correlation with. But the main conclusion of this is that regardless of socioeconomic status and irrespective of where people live, they're all exposed to similar concentrations of microplastics in the air, at least within a region as large as Los Angeles County, perhaps due to atmospheric dispersion. So now that I've kind of confirmed that microplastic contamination is everywhere, and it cannot necessarily be predicted like other particulate matter, let's actually try to understand the mechanisms of emission better. Do they act different than, say, soil colloids or other particulate matter? And that leads me to chapter 6. Chapter 6 tries to answer what are the causes of preferential emission of microplastics from biosolid applied agricultural soils. This paper was published in 2024 in ES&T Letters and got a fair amount of media coverage from like the LA Times, ACS, Viz.org, and other kind of popular press. The motivation for this work is that biosolids in agricultural soil can be a source of pollutants. So what are biosolids? Essentially, when stormwater or wastewater moves through wastewater treatment plants, a lot of it kind of settles out into sludge. And sludge is then concentrated, dried out, and sold as biosolids and used in agricultural fields. It's a significant part of the circular economy and a great way that we've tried to, recycle waste and make it more usable. But wastewater treatment plants are actually really, really good at removal of microplastics without even being designed for it. Usually they're higher than 90% effective. So these sludges, these biosolids, are heavily contaminated with microplastics. So when we apply them all over the topsoils of our agricultural fields within the United States of America, they can be emitted into the atmosphere. And so we really wanted to try to understand that link between biosolids and atmospheric emission, and perhaps a difference with other soil colloids. And so we predicted that perhaps microplastics would act different than a traditional soil colloid. So if you see in this graphic here, the brown dots represent a typical soil or sand colloid, and this kind of white paper looking thing represents a microplastic. Again, they move like other colloids, they can saltate, they can directly suspend, but they have unique properties, right? Some of these properties I talked about in the introduction. They have a low density and they have a high hydrophobicity, or repellence of water. And that means that, the liquid bridge effect could be reduced for plastic compared to other soil colloids. And this might lead to their preferential emission into the atmosphere. And this chapter really aims to explore that. So why do I even think that the low density of microplastics could affect their emission, their erosion? And most papers do focus on density as kind of an obvious way that microplastics differ from traditional colloids when looking at emission potential. Here, this is like an equation from Xiao and Liu 2000. It's kind of like the foundational study within this field. U star represents the fluid threshold. This essentially, you can just think of it as the wind speed or the shear wind speed necessary to emit a particle. And here you can see it relies on the density of a particle. So it is proportional to the density of the particle you're looking at. And sand and microplastics have very, very different densities. So sand has a density on the order of 2.65 to 2.8, whereas plastics density varies from about 0.9 to sometimes as high as 1.4, but somewhere within this range. And so potentially the lower density of microplastics, due to its influence on this equation, will reduce at a lower U star, a lower threshold fluid shear velocity, and hence higher erodibility. We also think that interparticle forces could control this, right? So this is an equation from Fisher 1926. It's also adapted by Dr. Ravi in 2006. And this equation has many variables, but the one I really want to focus on is this gamma here. This gamma represents the contact angle of the particle, which differs between soil and plastic. And for larger particles, mostly like over 100, the important force here, or the important topic here, is the gravitational force, right? That's what really controls emission of these particles. That's why their fluid threshold on the y-axis is so high. But when we look at these smaller particles here, that's where interparticle force plays a much larger role. So lower than 100 microns, interparticle forces play a larger role in controlling the fluid threshold or increasing the fluid threshold. But if those are reliant on the contact angle or the hydrophobicity of a particle, and plastics has a very different contact angle compared to other soil colloids, then naturally it is to think that the interparticle force, which binds microplastics to terrestrial surfaces, could also significantly differ. And we know that microplastics are more hydrophobic than natural particulate matter. So on the left side, this is that same density graph I already talked through. But here, if we looked at a contact angle for sand has a contact angle around, say, 35 degrees, quite low, whereas the contact angle for microplastics is much higher, quite hydrophobic, usually around 80 degrees and higher. Since the threshold shear velocity depends on these interparticle forces, which are dominated by moisture, microplastics with higher contact angles would likely have weaker interparticle forces and lower threshold velocities. And that's something that I kind of prove using this theoretical framework. And I'm trying to answer what are the causes of preferential emission of microplastics from terrestrial soils. So this is kind of that first result. So I adapted this equation from Xiao and Liu in 2000 and plugged in the new values for density. So rather than a traditional soil colloid, I'm now adapting this for plastic to see, and the other constants are coming from Koch in 2012 and Koch and Renault in 2006. And I'm create this kind of theoretical framework where you can see sand has a fluid threshold that's much, much higher than plastic at whatever, at a given microplastic size. And actually you can see this trend of PT being the highest here. That actually has the highest density, and as you go down to polypropylene, that has the lowest density. So you can see that the trend in fluid threshold is very reliant on density. It goes down as density goes down. And actually, when we look at this minima here, which represents, the microplastic size at which the fluid threshold is the lowest, or it takes the lowest wind speed to emit that particle, we actually define a critical size of max resuspension between 100 and 200 microns, which is relatively large. But I compared those results to other literature review later on in this presentation. Why do I think that the fluid threshold is much lower? Again, as I kind of discussed the motivation, I predict it's because the interparticle force is lower for microplastics. So again, we adapted this equation from Fisher and Ravi, which is reliant on gamma, and here I have the variables, the parameters that I plugged in, the contact angles for plastic, the 4 plastic types, they were quite hydrophobic compared to sand, which has only 35 degrees. Generally, the interparticle force of a particle is controlled by the liquid bridge. So here, I kind of have a diagram to help explain to you what the C and the B are in this equation. They're related to kind of the meniscus curve between 2 colloids, and the equations are above here, but the most important part of this equation is the reliance on interparticle force on the contact angle of a colloid. And when we actually compare the interparticle force between sand and microplastics or plastic polymers, you can see that there's actually an attractive force for sand, a positive interparticle force. But as the contact angle decreases, that force then becomes repulsive and is much, much lower interparticle force for plastic polymers compared to soil colloids. And so our theoretical framework predicts that microplastics have a reduced attachment force, a reduced interparticle force, and thus a lower threshold velocity, and that could mean their higher emission potential. So we verified this theory using experimental results. So we managed to take samples of the biosolids that were being applied to an agricultural field, as well as soil from that agricultural field, and dust that was collected from an isokinetic sampler at a wind tunnel. This study was performed in 2017, I believe. At the time, these results were being processed differently, so I'll kind of briefly go through how we quantified microplastics in these samples. We took the soil, which represented the soil or the biosolid or the dust samples. We used density stratification, vacuum filtration. Then we digested the samples to try to remove the majority of the organic matter. Again, vacuum filtrated, density stratified, vacuum filtrated, and then counted these microplastics using a compound microscope, and this was verified using Raman at the time. We later switched to FTIR, thanks to the Moore Institute. So the results of the actual experimental portion of this chapter is that we did find enrichment of microplastics in wind-blown sediments. And if you remember what I discussed from my theoretical framework, this is exactly what we expected. So when we quantified microplastics, we see that the concentrations in the wind-blown sediments or the dust were statistically significantly higher than either the biosolids or the soil where those dusts left from. It's quite interesting, also quite problematic. We then also characterized those microplastics using FTIR, and we found the fractions, between 50 to 100 and greater than 100 microns were actually the most predominant fraction. So the larger microplastics were more populated in dust samples. And when we compare that, that size range to the other studies, I think there are 21 other studies at the time that had characterized microplastics in wind-blown sediments. We see that the majority of them also kind of fall between that range, or include that range, that critical size of max resuspension between 100 to 200 microns, which just further verifies our theoretical framework, right? We're seeing that preferential emission. We're seeing that critical size of max resuspension be verified by experimental results. And why is that important? What are the implications of this work? So we also took 3 months of wind data at 4 different heights at a bare site, also in Washington. And when we plotted those wind speeds and then calculated the fluid threshold for both plastic and soil, using that Shao and Liu equation that I've already discussed a few times, we found that within that 3-month period, when we have a fluid threshold of 5.84, which was derived from a Duke's paper at that specific site, within 3 months, there were over 100,000 events that could emit soil from that site. However, When we now calculate the fluid threshold of 1.64 meters per second for microplastics, that brings the fluid threshold all the way down to here. And so now within that 3-month span, there was over 400,000 events which could emit microplastics. So we're missing, well over 300 events, which are capable of emitting plastic, but not other traditional soil colloids from a site. And so when we kind of look at the percentage of events, there's a 269% increase in the number of events which are capable of resuspending microplastics than previously predicted. This could help inform EPA's fugitive dust models, which are, reliant on traditional soil colloids and don't factor in kind of modern anthropogenic pollutants such as microplastics, which are a huge inhalation concern. And they miss, 61% of wind events within a 3-month period, which could emit those into the atmosphere. So kind of the take-home message of this chapter, data-wise, is that the microplastic fluid threshold is 1.64, which is significantly lower than 5.84 meters per second for soil. We confirmed by theory and experimental data that microplastics are indeed preferentially emitted from terrestrial surfaces. Kind of the implication of that work is that traditional fugitive dust emission models underpredict microplastic emissions by around 61%. And it's impossible to protect human populations from risk of inhalation if our current dust emission models aren't even factoring in the events which could emit microplastics. And that kind of moves me into the next section, my next chapter. So in chapter 6, I predicted that density and hydrophobicity cause lower inner particle forces, which results in preferential emission of microplastics. But let's kind of zoom all the way in and actually quantify these forces, which, these inner particle forces, which bind microplastics to terrestrial surfaces, and also look at the influence of environmental factors such as relative humidity or UV exposure. So this chapter 6 is a broad overview confirming preferential emission, and chapter 7 zooms into these inner particle forces. And we're trying to answer the question, how do, weathering of microplastics and humidity affect the adhesive force between microplastics and sand, which in turn affects their emission by wind. So again, what binds a plastic to a sand are these inner particle forces. Those are what prevent, shear wind from moving and emitting a particle at a fluid threshold. So we want to quantify these inner particle forces and quantify what factors could affect those forces. So what are the inner particle forces that act on microplastics? There's a wide variety of forces between two colloids. Some of the more popular ones discussed are van der Waals, electrostatic forces, capillary forces, and hydrogen bonding. But as you can see from this diagram from Harak and Barty in 2021, for smaller particles between 0 and 1,000 microns, which is what we're looking at primarily for microplastics, the capillary force, this meniscus force from the liquid bridge, is the greatest force. So we can just assume that the other ones are fairly negligible, and we're just going to zoom in and look at this meniscus force, this capillary force. So what are my theoretical predictions? First off, what I want to discuss is that for this paper, we used a fairly large AFM tip, which had a 5-micron silicon dioxide tip, cantilever tip that was attached to the cantilever. Usually these tips are on the scales of nanometers. So when we actually zoom into the nanometer scale, and I kind of come up with this mock diagram to show, it looks like a ball from all the way out here, but actually to the particle, to the surface of the sample at a nanometer scale, it actually looks like a plane. And so for this tip geometry from a Farsi and Tabrizia paper in 2008, you can quantify this meniscus force, this capillary force, which is the dominant interparticle force between plastic and sand, using this equation. And that equation is reliant on theta, which represents contact angle, which is 54 degrees for silica, which is the dominant mineral in sand, and 104.9 degrees for polypropylene. So a significant difference there. So we predict that this contact angle plays a large role. It's also reliant on P over P naught, which represents relative humidity. So that's the other variable that we're going to discuss in this chapter. contact angle can actually be affected by UV exposure. So as UV exposes on a sample on a plastic surface, it can kind of break all the bonds on the surface or distort their typical surface interactions and cause them to be either more hydrophobic or less hydrophobic, depending on their exposure. So the research question here are, what are the effects of UV exposure on contact angle and thus adhesion forces between microplastic and sand? And also, what are the effects of relative humidity on the adhesion forces between microplastic and sand? So kind of this relative humidity and this UV light are the two main environmental factors I'm exploring, and I'm quantifying this interparticle force, which I've proxied by just the meniscus force, as it's the dominant interparticle force for this interaction. So how did we measure the interparticle force? We measured it using AFM, atomic force microscopy, which is, pretty standard method for this line of work. AFM, essentially, for those of you who don't know, I'll just broad scale introduction. You bring down a cantilever or a tip close to a sample, you bring it all the way down just until touching, and then you retract it away. And what I've shown here is this is a typical force curve, a typical retraction force curve for AFM. And the minima and the difference between this baseline and this minima represents the adhesion force. So you can actually extract that adhesion value from each force curve. And so what we did is for every sample, a plastic sample or a glass sample or, a glass slide sample, we would tag the surface. So that way we could always return back to the same spot. And at a set distance away, we collected 4 force maps. So this is what a typical force map looks like, and each one of these mini squares represents a 1 by 1 micron, and it has a force curve associated. So each square essentially collects 100 force curves. So we collected 400 force curves in the same spot every time, despite changing the other variables, as surface, heterogeneity of adhesion forces is a pretty common problem. And the variables we looked at was the effect of UV exposure. So essentially, we looked at a polypropylene sample at 0, at baseline, no UV exposure, and then we irradiated it for 1 minute, 5 minutes, 15 minutes, and 30 minutes, and we looked at adhesion and how it changed over this UV exposure. And we also looked at the effect of humidity by analyzing both a polypropylene sample and a glass sample at 50%, 60%, 70%, 80%, and 90% relative humidity. So I want to briefly talk about how we manipulated humidity in this experiment. Kind of my little pride and joy, my DIY setup for this. The goal of this was to use only commercial equipment so it's easily accessible, and also to kind of rapidly change relative humidity in this small sample dish, within 5 to 10 minutes to adjust 10% humidity. How did it work? We had a commercial humidifier with a tube that you were able to kind of set the humidity. And then we linked that tube using a variety of reducers and silicone tubing to this sample, which was covered with a silicone, like a silicone cone, and just the AFM tip was allowed in. It had an inlet and an outlet, so the humidifier fed into the inlet, and we also had an outlet that led to an outlet chamber, which was a glass bottle with some holes drilled into the top, just tight enough to fit in the external sensor. And here's the external sensor, and in real time that outputted the temperature and the relative humidity within the system. So that way we could be sure that when we were taking measurements, it was at that humidity that we wanted, and that stayed stable. And what were the results of this study? So first we just looked at UV exposure and contact angle. So we just kind of wanted to figure out how is UV exposure affecting the hydrophobicity or the contact angle of a microplastic, of a colloid, because that contact angle is what the interparticle force or the adhesion force is reliant on. And what we found is we had about 20 data points per exposure time from 2 different samples. What we found is that an increase in UV exposure, so from 0 to 30 minutes, and this is in log scale, significantly decreased the contact angle of polypropylene. So it started, a little bit above 110 degrees, and the median for the most exposed time was around 90 degrees. So it becomes less hydrophobic over time. So it increases its hydrophilicity as UV exposure increases. And how does that change in contact angle now affect adhesion force? That's our next result. So again, looking at log scale for UV exposure between 0 and 30, but now on the y-axis I have adhesion force. And this was actually recorded by AFM. So per each measurement, I have 400 force curves, so 400 adhesion values that I'm extracting using R. And I found that an increase in UV exposure significantly decreased adhesion force. So you can see the median value kind of drops off, and it doesn't drop off super linearly. So you can see between 1 and 5, that's kind of that huge jump down. And once most of the adhesion force, falls away, it kind of stays fairly stable at that lower point. So because the adhesion force is lower for, more degraded, more weathered microplastics, that means more weathered microplastics could also be more likely to emit, right? There's less force binding them to threshold surfaces. So fluid threshold necessary to remove them will be lower. And now for our humidity experiment. So this graph depicts both polypropylene, so the box plots and dots in green are the plastic, and the purple is our control, which is just the silica glass side. And we found that increasing relative humidity between 50 and 90% decreased adhesion for both polypropylene and glass. So the effect was the same, adhesion fell. But the difference is that the effect was much more prominent for polypropylene. So you can see the drop off in adhesion happens much faster for plastic. So even within this first 20%, almost all the adhesion falls away, and that stays the same as you continue onward. Whereas for plastic, that decrease happens a bit more slowly. And, the major drop off happens after about 40 degrees. So it's a little bit of a slower decrease, and also the effect isn't as strong. So the net loss in adhesion force for silica was less than the net loss in adhesion for plastic. So humidity affects the adhesion on plastic more so than it does soil. So what's the take-home message of this chapter? A is that increased UV exposure and increased relative humidity both lead to a decreased adhesion force between plastic and sand. The majority of adhesion force is reduced after just 5 minutes of weathering. You lose 67.5% of adhesion force, and after a 20% increase in humidity, you lose almost 80% of the adhesion force. The drop in adhesion force is stronger for polypropylene than plastic. And what does that overall mean? Is that microplastics are more loosely held to terrestrial surfaces than sand, and that is even more true for weathered microplastics, which are exposed to a lot of UV, or in more humid environments where relative humidity is higher. And therefore, in these cases, microplastics could be emitted more easily into the atmosphere. And that's kind of the breakdown of all my chapters. I just kind of want to go through even the chapters I didn't present today, kind of just in my dissertation summary. It's all in my thesis, which I sent to you a couple weeks ago. But chapter 2 looked at land use and climate. So we didn't find an effect for land use, but looking at regional climate. So this map here is the Koppen-Geiger climate classification across the globe, and the little black dots are all the global literature that we analyze. And we found that a region's climate did play a greater role than land use in predicting the deposition rate of airborne microplastics. And we actually found, interestingly, higher concentrations in drier climates and more humid climates. At the time, we weren't quite sure. We thought in drier climates, of course, there's less liquid bridge, so microplastics could be emitted easier. But actually, chapter 7 kind of verifies that as humidity increases, so in these more tropical climates, adhesion force is lower. So that could also lead to preferential emission of microplastics. So chapter 7 kind of verifies our review paper from chapter 2. Chapter 3, I actually looked at a depth profile from a sediment core in the Gulf of Mexico. I found that most of the microplastics were trapped within the top 6 centimeters of the core. If we take a 3 millimeter per year deposition rate for the Gulf of Mexico from another paper, this kind of implies that there's been a rapid onset of contamination within the last about 2 decades. And that kind of correlates with our increase in plastic usage as a consumer product. Chapter 4 evaluated using the urban canopy as, a passive sampler for airborne microplastics. We looked at 3 different factors: the height of the leaf above the ground, the land use where that leaf was taken, and the leaf surface hydrophobicity. We found that height contributes most to variability, so where we sampled from, if I remember correctly, above 1.5 meters to about 2 meters was the best case to reduce variability. But I've also inserted this graph where you can see that the contact angle, so as the hydrophobicity of the leaf increased, we did see a slight correlation with decreased microplastic concentrations, though our R-squared wasn't very high. So height plays a large role, but the overarching takeaway from chapter 4 was that leaves introduce too much variability into reported microplastic concentrations, and so we just scrapped that idea entirely, and we now use the smartphone method discussed above. Chapter 5, as I discussed today, so I'll kind of zoom through these takeaways. Unlike traditional particulate matter, airborne microplastics may not be predicted based on proximity to the source or socioeconomic status of the region, so we aren't able to make a map like this. But it's important because this is the first kind of work in this field ever done, right? So it's the first work to kind of prove that, hey, perhaps atmospheric dispersion of microplastics plays a larger role than atmospheric dispersion of other particulate matter. And that's important to note. That's important for future risk assessments and things like that. Chapter 6, microplastics are preferentially emitted below the characteristic fluid threshold for soil, and that is attributed to their low density and their high hydrophobicity. Then chapter 7, we actually move into a microscale quantitative analysis, and we find that the interparticle force between a microplastic and the soil decreases with increases in humidity and weathering of microplastics under sunlight. And that's kind of an overall summary of the entire body of my work. Kind of future studies, takeaways from all my chapters. The majority of the methodology recommendations come from my chapters 2, 3, and 4. We see that if you're doing passive samplers to analyze atmospheric concentrations of microplastics, you should have a sample duration longer than 10 days in order to reduce variability. We found that concentrations of microplastics in seawater and sediments vary significantly based on detection limits, based on the reported detection limit. So if your detection limit is lower, meaning you're saying you can see smaller particles, your concentrations are higher. And that's important because you can't cross-compare studies then. There's a need for long-term passive or active samplers to kind of establish a baseline for microplastics, atmospheric microplastic concentrations. Another future study, something I didn't get to do as part of this thesis work, but I hope one day somebody gets to, is actually even further verify this preferential emission of microplastics in air by actually verifying the density of microplastics and the hydrophobicity of plastics and seeing how they emit, collecting them into an isokinetic sampler and draw a conclusion based on that. Overall, that will help to further develop accurate fugitive dust emission models. Lastly, I think there's more force work that can get done. We could evaluate either the combined effect of weathering and humidity on adhesion force. Right now, we kind of looked at those variables separately, but what is the combined effect on emission potential? Another thing that we could do is we looked at humidity ranges between 50 and 90%. You could actually look at 0 to 50% relative humidity, evaluate super low humidity conditions. That could be important because as climate change kind of continues, there will be a rise in aridity of regions, an exacerbation of aridity in regions. And so it's potentially important to understand how emission of microplastics will also change with that climate change driven aridity. And then something I would have loved to do, as Dr. Mohanty kind of mentioned at the beginning, I'm really, really interested in kind of the health, the risk of, atmospheric inhalation of microplastics. But I think we could kind of combine this work using AFM and quantify the interparticle force using a plastic tip. So we use a silicon dioxide tip to represent sand, and we use a plastic sample to represent the interaction between sand and plastic. But actually we flipped that and we use a plastic tip, and then we took a live lung cell and we looked at the interactive force between those two. We could quantify the interparticle forces which would A, help us understand the transport within lung tissue, and especially understand why perhaps larger microplastics are found deeper in the lungs, where we don't necessarily expect them to be. As well as you can use methods like fluorescence lifetime imaging to measure oxidative stress within a lung cell based on, say, the plastic properties, so which plastic type of the tip, or the length of the contact, or how many times you touch the lung cell, or something like that. Yeah, and this is the entirety of my presentation. Before I end, I really want to say a deep thank you to my committee, Dr. Mohanty, my chair, and my advisor, Dr. Mahendra, Dr. Hawk, Dr. Jasby, and Dr. Cowger. Thank you all for being here today and being so flexible with remote for my health. I really appreciate that. I also want to say a special thank you to everyone at C-Lab, but a special thank you to Dr. Kutnik and Dr. Porthaker, both who I collaborated with significantly as I was kind of coming into my PhD, and I attribute a lot of my success to them. And to all my collaborators, so Dr. Koitamir, Dr. Sang, and Dr. Ozkan, thank you for their help in developing the smartphone method, which all of this work was published with. To Dr. Ravi, who's here, who was on my committee for my prospectus and has been a huge help in, mentoring me through all of these equations for atmospheric emissions, which is his expertise. To Dr. Reagan Patterson, who helped a lot with the socioeconomic paper. And lastly, to Michael Lake and Dr. Adam Steeg at CNSI, who helped us with the AFM work. And to all my funding sources, NSF, UCLA Civil Engineering, NOAA Sea Grant, and of course, the Moore Institute for Plastic Pollution Research, who have very gracefully let us use their FTIR machine for free for like the last 4 or 5 years. And none of this work could be possible without verifying our results using FTIR. So yeah, thank you so much.